Ladies and gentlemen, please welcome back Jennifer Scott Williams.
Okay, come on, applause. Look at this. No. Let's get this thing going. Welcome back. We hope you all enjoyed the technical sessions and the variety of scientific research and development covered during those sessions. The ISS National Lab has a rich history of partnering with some of our leading scientific and funding agencies within the United States. The National Science Foundation is one of those tremendous partners. For the past 7 years, the National Science Foundation has partnered with ISS National Lab to fund and further transport phenomena and fluid flow investigations on the orbiting platform. Additionally, the past 5 years have seen solicitations focused on tissue engineering concepts in a space-based environment. This session will allow the audience to learn a little more about why the NSF sees value in continuing fundamental research on the space station, and you'll get to meet some of the investigators who have launched research to the ISS through NSF funding. I'd like to introduce our moderator for this session, Stephanie George. Stephanie is the Engineering of Biomedical Systems Program Director for the NSF. She manages a broad portfolio of science and engineering, from tissue engineering to computational modeling of physiological processes. Please welcome Stephanie George and her panelists to the stage.
Thank you for the introduction. The microgravity environment of the ISS provides unique opportunity to explore fundamental engineering questions to ultimately benefit life on Earth. The National Science Foundation was one of the first government agencies to recognize the value of the space-based research and development. As mentioned, NSF has partnered with the ISS National Lab through cases on 2 long-standing solicitations. The first, transport phenomena within the Division of Chemical, Bioengineering, Environmental, and Transport Systems, or CBET, was established in 2015, and the tissue engineering and mechanobiology solicitation began in 2017 and spans 2 engineering divisions, CBET and CMMI, the Civil, Mechanical, and Manufacturing Innovation Division. These exciting projects enable scientific discoveries not possible on Earth. But what I think is truly exciting are the partnerships that have been developed. In fact, partnerships are a top priority for NSF leadership, and as noted several times today, important for the ISS. These solicitations represent a successful partnership between government, academia, and industry. All 3 partners are needed, as no one entity could do it alone. These solicitations have brought together a community of scholars passionate about leveraging the unique environment of the ISS. This requires engineers to think creatively and optimize for this design space, which these PIs can probably attest is not an easy task. Research teams interact with astronauts who carry out the experiments on the ISS. What an amazing opportunity for researchers and the trainees involved. Our panel today highlights the work that has been or will be done in the microgravity of low Earth orbit and the value of our NSF, CASIS, and ISS National Lab partnership. I'd like to introduce the members of the panel and have them share a little bit about their research. Our first speaker is Dr. Tammy Chang. She's an associate professor of surgery at the University of California, San Francisco. She holds an MD, PhD combined degree from Harvard Medical School, and her clinical training includes general surgery residency and minimally invasive surgery fellowship at UCSF. Dr. Chang attends on the acute care surgery service at the UCSF Medical Center, and she directs a basic and translational research laboratory focused on liver tissue engineering. The unifying theme of her research program is to understand how physical environmental forces, including dimensionality and extracellular matrix, regulate hepatocyte function. Overarching goals of Dr. Chang's laboratory are to develop novel tissue-based therapies to treat end-stage liver disease and to advance bench to operating room regenerative surgery approaches. Thank you for joining us, Dr. Chang.
Good afternoon. Here we go. So when I think about let's see. All right. So when I think about why I do the research that I do, I think about this graph. This is a graph of the number of transplants and the number of people waiting for transplants in the US for any organ for the past 25 years. Transplant is the only definitive treatment for end-stage organ failure, which is the fatal, irreversible dysfunction of a vital organ, and it affects millions of people worldwide. Over the years, the number of transplants have increased, but the number of people added to the transplant each year has increased even more. The result is this line on the top, which is the number of patients who are still remaining on the liver transplant list at year's end. The reason why there's such a gap between the number of transplants that are performed and the number of people who need transplants is that the supply for donor organs is limited. This has contributed to thousands of people dying on the transplant waiting list before having the opportunity to receive a transplant. However, this data is an underrepresentation of the true need because many people who may benefit from a transplant are never listed. So when I look at this graph and the trends of these lines and the magnitude of the gap, what it says to me is that the answer to end-stage organ failure has to be something other than more transplants. So of all the vital organs, why the liver? It is because I believe that finding novel approaches to replace the liver function has the most dire need. The heart is a magnificent organ, but really it has one function, and that is of a pump. And the biomedical community has figured out how to make a pump. It's called the left ventricular assist device. It is implanted, people walk around with it, it can be a bridge to transplant, or it can be permanent therapy. The lungs. The lungs are essentially gas exchange membranes. And the biomedical community has developed a system called the Extracorporeal Membrane Oxygenation, or ECMO. During this pandemic, the sickest patients are put on ECMO machines to help breathe for them while their lungs recover. And what about the kidneys? Well, the kidneys are basically a filtration system. And our ability to replace that filtering function dates back to the 1940s. People who have non-functioning kidneys can go to a dialysis center 3 times a week and live for years. What about the liver? For the liver, we have nothing. If you suffer a catastrophic event and your liver shuts down completely, you need a transplant. That's it. We don't have a machine that can tie you over even for a little bit. Why? Why is replacing liver function such a tough problem? It is because the liver has more than 500 vital functions. All of them are biochemical reactions of metabolism and synthesis. that are in dynamic feedback loops, all coordinated and cross-regulated. How are we going to do this without the cell? The living cell, perhaps the most amazing machine of all. It has stood the test of time. It's been forged by billions of years of evolution. So I would posit that to develop a novel approach to replace liver function, we need the living cell or the living tissue as the fundamental workhorse. And while the need to replace liver function is the most dire, the use of cell and tissue-based therapies for the liver is also the most promising. And that is because the liver has an innate ability to regenerate. If you have a healthy liver, 2/3 of it can be removed, and the third that is left will grow back to the exact size you need to fulfill your metabolic demand. And somehow the Greeks knew this. So as a punishment for giving humans the gift of fire, Zeus tied Prometheus up to a mountain and sent an eagle to eat his liver. Now, this punishment is cruel because every night Prometheus's liver would grow back. And every morning, the eagle will come and attack his liver again. So in a normal liver, it is the mature hepatocytes that's doing the regenerating. In some circumstances, liver-resident stem cells can play a role. For novel tissue-based therapies, our lab is focusing on induced pluripotent stem cells. These cells are adult cells that have been reprogrammed to become stem cells, which then can be differentiated into liver cells. We believe that this approach is the most versatile and the most scalable. Stem cells can be used to form organoids, which are three-dimensional aggregates of organ-specific cells. As a surgeon, our lab is primarily interested in developing liver organoids for implantation therapy as an alternative to transplant. However, there are multiple potential applications for liver organoids, including drug development and testing, precision medicine diagnostics, disease modeling, and use in extracorporeal liver assist devices. Now, the technology has not matured to the point of fulfilling these potential applications. And that is because terrestrial methods of making organoids produce organoids that only have a fraction of the function of a real hepatocyte. Terrestrial conventional methods depend on matrigel, which is an extracellular matrix produced by a mouse cell line. Now, the fact that this component comes from a different species makes it challenging to translate for clinical therapy. In addition, matrigel is incompletely defined, and there is significant batch-to-batch variability. And these issues make reproducibility, mechanistic research, and quality control problematic. Moreover, the approach is low throughput. Our lab has shown that stem cell-derived liver cells can self-assemble into organoids in simulated microgravity without the need of matrigel, as opposed to matrigel-based methods that form one organoid per culture well. Numerous organoids are developed in a single microgravity suspension culture. And so the microgravity approach is higher throughput. But importantly, matrigel organoids, their 3D structure degenerates through time, whereas the 3D organization of organoids in microgravity are sustained. Not only do organoids in microgravity have improved form, importantly, they have improved function. Albumin production is an important hepatocyte function, and microgravity organoids produce more albumin per cell than matrigel organoids. In RNA-seq, RNA sequencing transcriptomic analysis, over 250 hepatic-specific genes are upregulated in microgravity organoids compared to cells differentiated as monolayers through established standard protocols and in comparison to matrigel organoids. These upregulated genes in microgravity represent and encompass a wide and diverse range of hepatic functions. So one can make the business argument that large-scale, high-throughput production of highly functional organoids for a multitude of biomedical applications has the economies of scale on its own merit to make biomanufacturing in low Earth orbit worthwhile. I believe that the low Earth orbit microgravity environment can provide an even more unique advantage. Organoids that are developed in simulated microgravity on the ground are small, and they stay small. There is evidence that in the microgravity environment of low Earth orbit, these organoids can further coalesce and form larger clusters. We hypothesize that with microgravity-enabled tissue self-assembly and giving discrete biochemical and mechanical cues, we can develop a large, vascularized, highly functional liver tissue from stem cells, a feat that is not currently achievable by Earth-based methods. Through computer modeling of particle distribution in microgravity, we conceived that the most ideal geometry for promoting organoid distribution and tissue self-assembly in microgravity is a spherical volume. This is an early concept drawing of the spherical culture vessel that we designed, and that will fly to the ISS in January, this coming January, to test our ideas on tissue, on microgravity-enabled tissue self-assembly. We have filed for both US and international patents for this device design. In conclusion, there's a critical need for novel tissue-based therapies to treat end-stage liver failure. Microgravity-enabled tissue self-assembly produces highly functional liver organoids. And low Earth orbit may provide a unique environment for large-scale, high-throughput production of stem cell-derived liver tissue for therapy. The work I presented were led by two talented scientists in the lab, Yun Wen, who is a research specialist, and Maria Secchi, who is a postdoctoral scholar. I'd also like to thank my collaborator at UCSF, Tejal Desai, and our implementation partner, Space Tango. And I also thank NSF and NASA for funding this work and other funding sources for the lab. Thank you.
So we are holding all questions to the end, and if you do want to submit a question, you can do that through the app. So thank you, Dr. Chang, for that talk. Next up, we have Dr. Nang Wang. She's an associate professor in the Department of Cardiothoracic Surgery at Stanford University and a principal investigator at the Veterans Affairs Palo Alto Healthcare System. Dr. Wang completed her BS in chemical engineering from the Massachusetts Institute of Technology, followed by a PhD in bioengineering from the University of California, Berkeley, and University of California, San Francisco joint program in bioengineering. Prior to joining the faculty, she was a postdoctoral scholar in the Division of Cardiovascular Medicine at Stanford University. Her laboratory investigates the interactions between stem cells and extracellular matrix microenvironment for engineering cardiovascular tissues to treat cardiovascular and musculoskeletal diseases. Recent research focuses on the role of microgravity for drug screening of engineered muscle tissue. Welcome.
Thank you for the introduction to speak. Today I'll share with you our exciting research that was launched in the NG16 launch just last summer. So we are a tissue engineering lab, and we seek to create tissues that can replace those that are diseased or otherwise dysfunctional. And for the past decade, we have been engineering skeletal muscle tissue for treating traumatic muscle injuries. With this recent NSF Cases Award, we sought to expand that research to see if we can use our engineered muscle platform to study important aspects of sarcopenia. So first, what is sarcopenia? Well, sarcopenia is really a global geriatric syndrome that's characterized by muscle wasting or muscle atrophy. And from the age of 20 to the age of 80, there is supposedly a 30% reduction in muscle size as well as in muscle mass. You can appreciate from the image on the right, which shows cross-sections of muscle tissue from young muscle as well as those who are old and sarcopenic. And you can appreciate that the cross-sectional area of those muscle fibers are much smaller compared to those in the healthy young muscle. Sarcopenia is also associated with frailty as well as poor health outcomes. Importantly, there are no existing FDA-approved drugs currently to treat sarcopenia. Mostly, doctors would recommend some supplements maybe, and maybe exercise. But one of the challenges for developing drugs to combat sarcopenia is that sarcopenia is a progressive, slow process. And so a company that would be interested in testing drug targets may have to spend 1 or 2 decades to test the efficacy of a drug target, which could be very costly and time-consuming. And so that's where we had this idea that, well, we know microgravity accelerates the process of aging as well as muscle use and disuse rather, and we know this from studying the astronauts that return. And so we thought, well, maybe we can apply our engineered muscle platform in microgravity to see if it can mimic certain aspects of sarcopenia. Because if it does, it could be a useful platform for potentially screening of drugs in a relatively short period of time compared to on Earth, which could take decades. And so we hypothesized that engineered muscle in microgravity mimics salient aspects of sarcopenia and that it could be a robust platform for testing and validating drugs to treat sarcopenia. So how do we create the engineered muscle? Well, we tried to create engineered muscle that mimics the structural physiological organization of native skeletal muscle. And we can kind of think of muscle as being a parallel bundle of myofibers. So to create that kind of spatial organization, we developed nanopattern collagen scaffolds that have nanoscale topographical cues on the order of about 100 nanometers. And when human muscle progenitor cells are seeded onto those scaffolds, they sense those cues almost like railroad tracks and will align their cell bodies along the direction of those nanopattern collagen. And so when we then induce them to form these multinucleated myotubes through a process called fusion, they essentially can create parallel bundles of myofibers. And so our scaffolds are designed in a geometry that is long and thin, and we kind of bundle 8 of these strips of collagen scaffolds onto immobilized glass slides that are seeded with primary muscle progenitor cells. And so shown on the image on the bottom, the muscle progenitor cells align their cell bodies along the direction of the nanopattern collagen, which is denoted here by the horizontal direction. And when we coax them to form these premature myofibers called myotubes, those myotubes also develop that same kind of parallel organization and express proteins such as myosin heavy chains so that you can visualize the organization of those myotubes. So that's how we created the engineered muscle. But we were really new to the area of space research. And so we learned a lot along the way. It was a steep learning curve, but luckily our implementation partners, Bioserve Space Technologies, really helped us along the way. And the first thing we learned in translating our research to space was adapting our engineered muscles to microgravity. So Bioserve Space Technologies worked with us to create compatible bioreactors for our engineered muscles. And our engineered muscles are immobilized onto glass pieces, I would call them. These glass pieces are screwed down into these bioreactors as shown by the yellow arrow, and then basically immersed into cell culture media. And on the right is an image of one of the space astronauts conducting experiments using these bioreactors. Well, to get ready for launch, we actually had a number of different optimizations to do, and those including making sure our bioreactors are robust and withstand different kinds of fluctuations. These fluctuations include that of temperature fluctuations, where our bioreactors were cultured in plate habitats or PHABs that were really good at maintaining their temperature. So this is a temperature trace of our launch samples from the time of handover as well as the time of launch, where we can see on the y-axis that the temperature really didn't fluctuate too much over time, no more than about 1 or 2 degrees. We also had to ensure that our engineered muscles could withstand the absence of media and oxygen exchange during the transit, which we projected would be anywhere between 4 to 7 days, and it turned out that it would only end up needing to be 4 days. We also had to make sure that our bioreactors could withstand shaking and vibrational motion associated with launch, and luckily we had shaker bioreactors, shakers that could help us simulate that. And of course, we had multiple bioreactor backups in case of a delayed launch. Well, so this was our experimental design. During the ground studies, we cultured primary human muscle progenitor cells on our spatially nanopatterned collagen scaffolds. And when reaching the station, they would then be switched to an induction media that induces these progenitor cells to fuse together to form these parallel aligned myotubes for a period of 7 days. And since we know that in sarcopenia, sarcopenia is characterized also by impaired muscle regeneration or impaired muscle formation, one of our major outputs was to determine in microgravity whether or not these muscle progenitor cells could form muscle myotubes in microgravity compared to regular ground conditions. And because this was a one-time experiment, we utilized primary cells from 4 different donors. And so I'd like to share with you some of our early data. We're still in the process of analyzing the transcriptional and proteomic analysis of our samples, but by and large, we do see important effects of microgravity. From just looking at donor-specific effects of this one particular donor, we can see that between the flight and the ground samples, there were over 400 genes that were expressed only in flight and over 500 genes that were only expressed in ground. And when we perform detailed gene set enrichment analysis, we see that many of the genes are actually downregulated in microgravity. And those genes that are downregulated with microgravity were associated with structural genes, structural muscle-related genes like myosin filaments. There was also a downregulation of genes associated with extracellular matrix remodeling, as well as growth factor signaling. But when we look at the cumulative effects of all of the donors, we see a quite interesting effect. And namely, if we look at just the top 20 differentially expressed genes, we're actually able to see some genes that were really interesting to us. So, and 2 of those genes are Notch2, and the other one is Perlecan. And these genes were actually both downregulated in the presence of microgravity. So Notch2 is a receptor that is normally associated with muscle progenitor cell proliferation and maintenance and renewal. And so it was interesting to see that Notch2 was significantly downregulated in the microgravity samples. And similarly, HSPG2, which is Perlecan, a basement membrane extracellular matrix protein that is normally transiently upregulated during the process of muscle fiber formation, was also downregulated. And so these were giving us clues that in the microgravity setting, we were beginning to see a signature that mimics that of sarcopenia. Now, to test whether or not this platform could be used in the future for drug screening, in our experiment, we also performed a pilot study where we utilized drugs that are already known to have positive benefit on muscle regeneration, and one of them being IGF-1, or insulin growth factor 1. And it's a growth factor that's necessary for muscle regeneration, but is known to be decreased with aging. And so in these experiments, at the time of the day 0 on cessation, the drugs were spiked into their induction media. And for 7 days, we also characterized their transcriptional changes. And so these results are still fairly early, but we are seeing that IGF does have differential expression of over 1,000 different genes, and we're still in the process of trying to analyze what is the relevance of these genes and whether or not IGF is able to at least partially reverse the effects of microgravity on muscle function. And so with this, I'd like to conclude by saying that we've demonstrated that our engineered muscle platform in microgravity was able to mimic certain aspects of sarcopenia. And importantly, this platform may be a viable platform for performing drug screening in the future. We anticipate in the drug discovery process that screening of drugs in microgravity could potentially serve as an intermediate step before the top hits or the top targets can then be further developed on Earth. So with that, I'd like to acknowledge my trainees as well as collaborators for their collaboration, as well as NSF Cases Award. And I would also like to take this opportunity to put in a plug that we have an upcoming launch in 2024 where we will be doing a larger drug screen. And so for those in industry who are interested in particular drugs of interest that will be relevant to our muscle platform, we'd be very interested in talking further to see if those could be drugs that we would test in our drug screen study in 2024. I'd be happy to take your questions afterwards. Thank you.
Great, thank you. So moving on to our third speaker, Dr. Eric First serves as professor and department chair of chemical and biomolecular engineering at the University of Delaware. Dr. First received his BS with university honors in chemical engineering from Carnegie Mellon University and his PhD from Stanford University. Prior to joining the faculty at Delaware, he studied biophysics at Institut Curie in Paris. His research interests span a range of topics in soft matter science and engineering, but focus in particular on colloid science and rheology. He recently co-authored the book Microgravity for Oxford University Press. Thank you for being here.
Thanks, Stephanie. The nanostructure of a material can be used to control the flow of energy through it. Depending on the length scale of that nanostructure, you can control a variety of different forms of energy: mechanical energy in the form of sound, thermal energy in the form of heat, electromagnetic energy in the form of visible and non-visible wavelengths of light. This ability to control the flow of energy gives rise to interesting potential technologies in optomechanics, devices like thermal diodes, and even the idea that maybe we could create some form of cloaking device if the material is structured in a specific way. So these obviously have some really interesting aerospace applications and space applications. If you're able to control the thermal conductivity of a material, for instance, by controlling the nanostructure of that material. What we've been investigating and what we've been interested in is how to build that nanostructure without sort of top-down typical manufacturing processes like semiconductor fabrication, where you're adding and removing layers of material to form that very ordered structure. Instead, we want that structure to organize itself, to self-assemble. That may seem fantastic, but in fact, the image to the left shows one example of a self-organized nanostructure. These are dumbbell particles, little fused spheres of polystyrene, basically, that have formed a crystalline structure. And that crystalline structure has properties like a Bragg diffraction property with it, so that certain wavelengths of light or even phonons of sound can propagate through it and others cannot. Now, one of the limitations of this approach is one of kinetics. In fact, what enables that is the fact that all of these little particles have thermal energy. If you put them in a fluid, they move around by Brownian motion. And because of that, they can explore configurations. They can rearrange with each other in a way that follows the laws of thermodynamics and finds a minimum in their energy. That's what gives that self-assembled structure and what allows us to form these really highly ordered arrays. But there's also kinetics, and sometimes you get trapped in a bottleneck, in this case, a glass, as shown in the far left image in this slide. Those particles, as they concentrate, lose the ability to move around because their neighbors impede them, right, from rotating and translating around to find the lowest energy state, the global lowest energy state. So you need some strategies that allow you to circumvent these kinetic bottlenecks. And we've been exploring the use of external fields to direct that self-assembly, to nudge it in the right direction without losing the ability for the material to self-assemble, to form its own nanostructure. Some recent theory from Jim Swan and his colleague, Zach Sherman, gave us some really interesting insights, and their results are shown on the right of this slide, where they showed that by using an external field and turning that field on and off, toggling it on and off, so that you change the attractive interactions between particles, actually allowed you to circumvent these kinetic bottlenecks, to tune the structure into one that can be predicted by the equilibrium thermodynamic models that they derived. And you can see some images from their computer simulations in the lower right of some of those structures that correspond to that phase diagram in the center. Their simulations and their theory were based on some of the earlier experiments that we did, so we have some nice back and forth in this, but it gave us exactly a hypothesis to test, which was that they predicted that the duty ratio, that is how long you turn a field on versus how short it is, and the frequency should matter in terms of deciding and determining where you're going to end up on this phase diagram. So that was the experiment that we went and tested on our recent space station experiments. We flew, this is the last in a series of experiments since about 2008 that we flew last summer, and you're gonna see some of the previews of the results that we got. We're still in the process of working on these. This is the experiment that we do on the space station. So in order to turn an interaction on and off using an external field, we use magnetic colloids. These are about a micrometer in dimension. They're all uniform in size. They have a little bit of iron oxide nanoparticles embedded in them, and that allows them to be paramagnetic. So in an external field, in a magnetic field that we apply, those particles will polarize and they'll attract one another, just like tiny magnets on your refrigerator door. In the space station, we have a set of Helmholtz coils that generate that magnetic field. It can generate it at a certain frequency. It's just turning the field on and off, generates it at a certain frequency, and we can control the duty ratio, how long that field is on versus how long it's off. You can see on the left a few images of the experimental setup, which is fairly straightforward in the Microgravity Sciences glove box. The Helmholtz coil down in the left with a sample sitting in it, and two camera views that allow us to look along the field direction and perpendicular to the field direction. And that gives us an idea of the microstructure which is forming. It allows us to see whether that material really does phase separate into a concentrated regime of particles with fluid surrounding it, or whether it doesn't, and what kind of structures it's ultimately going to form. So those images, you can see some sped up videos from what we call the ST camera, the straight view. That's looking down the field direction of our magnetic field, and perpendicular, which we call the RT camera view. And what we found was very interesting structures throughout our experiments. So why do we do this in space, I think is a question that's especially important to address here in microgravity. If you're looking at thermodynamics and thermodynamic processes, the time scales to reach equilibrium, especially for these larger particles, which are convenient to use for our study, right? Because we can see them, we can use microscopy to study them, the time scales are such that we can capture them with video cameras. It makes it slow, right? And it makes it slow enough that our experiment takes a little over an hour to perform. So it's beyond what we can do with a drop tower. It's beyond what we can do with sounding rockets, with parabolic flights. We really need that environment of the space station to do the experiment in terms of a long duration microgravity experiment. And it doesn't hurt to have hands on the experiment as well. We've really partnered and collaborated with the astronauts over the years and the crew over the years to really get the most science out of our experiments. But these particles, because they have iron oxide in them, are heavy, and they sediment fairly rapidly, especially when you place a magnetic field on and aggregates of large size form. In fact, you can show that in 3 dimensions, no matter how carefully you try to density match that, which you can't do with these particular materials, but you can come up with some other strategies, the size of the structure that's forming really prevents you from, on those long time scales of our experiments, preventing them from sedimenting at all. So this is why we need the space station to do this work. Now that sedimentation shouldn't affect the particles as we go to smaller and smaller sizes. But again, we pick these particles because of the convenience of the experiment, basically, because we can see what happens. And this slide summarizes the experiments that we returned with last year's experiments. We had downlinks with the initial images of the data, and we could see what the astronauts were doing, but it took some time to get the SSD drives downmasked from the ISS, and we're now working on trying to understand what we saw. What was exciting about the experiment that we did is we saw some very interesting structures, which we had never seen in ground-based experiments before because of that sedimentation effect. So these images are all sped up. This is an hour of experiment in each of these images that's going by, and you can see the structural evolution of our suspension with time. Now, that evolution and that structure which is forming depends on, as we expected from our hypothesis, the duty ratio and the frequency of the field. If you turn the field on too fast, on and off too fast, structure doesn't relax that well and it never equilibrates. So in the bottom left of this slide, you can see this sort of columnar structure evolve. We're looking down what we think are columns forming in this, where the columns are aligned with a magnetic field, and that structure really doesn't coarsen or change much with time because it's essentially frozen by this high frequency. But if you go way over to the right of our phase diagram, where we're at low frequencies, basically, no visible microstructure can form because the field is off too long. In the sweet spot, somewhere in the middle of frequency and duty ratio, we see some really interesting aggregates and structures start to form in our experiments. We find anisotropic structures begin to form at modest frequencies around the middle of our phase diagram and at higher duty ratios. But what was really exciting is this top right image that you're seeing. These very dynamic sheets that seem to form. Well, we think they're sheets, that they're propagating into the field direction, but we had never seen anything like this before, where you can see this large anisotropy in these materials start to form and exchange material from one aggregate to the other. So there's a tremendous amount of dynamics in this as well that are really exciting for us. That was what I wanted to tell you a little bit about in terms of our experiment, what was motivating it, and what our results look like here. And I wanted to end by just thanking a number of people because there are so many people that contribute to experiments like this. My postdocs and PhD students listed at the top, a number of undergraduates that participated in our laboratory with these experiments. Jim and Zach Sherman, who I mentioned before, Zintech with their colleagues that helped us perform the experiments last summer, and of course the ISS crew and the NASA folks at Glenn, Johnson, and Marshall Centers who kept everything together. So thank you for your time today, and I'm looking forward to the questions later. Thanks.
Thank you, Dr. First. So our last speaker is Dr. Yating Liao, is an associate professor and the faculty director of the UL Fire and Combustion Laboratories at Case Western Reserve University. She also heads the Computational Fire Dynamics Laboratory in the Department of Mechanical and Aerospace Engineering at Case Western. Dr. Liao's research projects range from basic fire science, for example, material flammability or microgravity combustion, to understanding fire behavior in specific situations, such as structure, wildland fires, or spacecraft. Her work has been supported by NASA, CASIS, NSF, and UL. Many of her projects involve microgravity experiments. She supports NASA's Space Project Sapphire, where experiments of large fires are performed in unmanned ISS resupply vehicles. She is also the PI of a project supported jointly by NSF and CASIS, where burn experiments were performed aboard the ISS. Thank you for joining us.
Thank you, Stephanie, for the nice introduction. Yes, our ISS projects are confined conduction. So in this project, we study how solid materials burn in confined spaces. Or to be more specific, we study the interactions between flame and surrounding walls in microgravity. Yeah. We know that fire behaves very differently in open and in confined environments. The reason is because in fire, thermal expansion occurs, and when subjected to confinement, the expanding gases are forced to accelerate in certain directions. And on top of that, the confinement can impose radiation reflection back to the flame or back to the burning samples. So these effects can result in very hazardous fire scenario. That is why in this project, we aim to examine the aerodynamics and thermal effects of confinement on fire behaviors. And we would also like to determine the outcome of flames in different confined conditions. So in this project, of course, we conducted microgravity experiments, which will be the focus of this presentation. But I also want to mention that we conducted numerical simulations and ground tests to complement the ISS data. So why microgravity? Yeah, the reason is because on the ground, we know that fire is hot, so it's always associated with buoyancy flow. The problem about the buoyancy flow is it is very messy. It varies with time and location as your fire grows. This flow is very hard to characterize, and it masks the underlying physics we would like to investigate. That is why we brought our fire to space. After the buoyancy flow is eliminated, we can impose an external flow in a controlled manner. This allows us to investigate each parameter independently. It also facilitates the interpretation of the experimental outcome. Okay, so the figure on the right explains where our experiments took place. Aboard the ISS, inside the MSG, the Microgravity Science Glow Box, we have a small wind tunnel like this size. My collaborator always says toaster size. And then inside the wind tunnel, we can impose a flow where our sample burns. Yeah, so and then, yeah, so you can see it's very safe. So don't worry, we are not going to set a fire accidentally on the ISS. We have a wind tunnel and everything's inside the glow box. So I'm the PI, principal investigator of this project. My co-investigator is Dr. Paul Foucault from USRA and NASA Glenn Research Center. The test hardware, the wind tunnel, was actually from a previous NASA project. It is called BAS, Burning and Suppression of Solid. And our implementing partner was Zinc Technology. And of course, this project was sponsored by NSF and CASIS. And our hardware was launched by CRS-19 in December 2019. So we really did very minimal modification of the hardware, the BAS wind tunnel. Zinc Technology first helped us to refurbish the wind tunnel, make it nice and clean. And then they help us to design and manufacture a new baffle sample system, as shown on the leftmost figure on the slide. So as you can see, our solid sample was burned between 2 parallel flow baffles. And by changing the position of the baffles and by using different type of baffles, we were able to simulate different level of confinement and different radiation boundary conditions. And then in this project, we burned 2 different type of materials. The first one is a PMMA, a type of plastic, thin plastic. And then we also burn a thin cotton-based fabric. We call this SIBO fabric. So to summarize, our parameter includes the interbaffle distance, or you can say confinement level, and the flow speed we impose in the wind tunnel. We also change baffle surface radiation properties, and we have different sample materials. So the figure on the right further explains our experimental setup. The wind tunnel, as you can see, features 2 observation windows, one on the top, one in the front. And then in our tests, we have a video camera looking at our burning sample from the top window. Later, I will show a few images of our flame taken by this video camera, the top video camera. And on the left figure, you can see some key personnel in this project. Again, the rightmost gentleman, that was my co-I, Dr. Pofaku, and the leftmost person is Chuck Bono, Mr. Chuck Bono, and then next to Dr. Pofaku, we have Ms. Emily Greenfink. They both are from Zinc Technology. I want to give them a big shout out because they've really provided excellent and tremendous support to this project. Without their help, this project could not have been so successful. Oh yeah, by the way, these pictures are taken at the Zinc Technology. They have a MSG mock-up there, or replica there. Right, so this slide explains how we communicated and worked with the astronauts during our ISS experiment. As you can see on the left figure, the science team and the Zinc Technology team sit together next to each other in the Telescience Support Center at NASA JRC. And okay, it's hard to not being able to point to things. But anyway, the second person on the left, his name is Dr. Michael Johnston from NASA Glenn Research Center. He was the project scientist for Confined Conduction. Right, so back to the operation. So Chuck has the coolest job during the operation. He was in charge of the space communication. In other words, he talked to the astronauts. And Emily coordinated the ground communication. That includes talking to Marshall and the Johnson Space Center. And we watch astronauts doing the experiment and also watch our flame on the 4 monitor you see on the middle picture there. So everything is real time, although they are like 250 miles above, above our head. So this is really neat because this allows us to make real-time decision for every single test point. This is really helpful. And then here are some example flame images. Yes, since we only have 10 minutes, so I'm so sad I cannot share everything with you guys, but here are some example results. So these are results for the cotton-based fabric. As you can see, while we vary the impulse flow speed and the interbaffle distance, and by using different flow type, we observe 3 different types of burning events. First, on the lower left corner, we have no spreading, meaning the flame extinguished after ignition. And on the middle region, we have steady flame, meaning we observe flame with a constant flame length spreading at a constant rate along the sample surface. And then on the upper right corner, we have growing flame, meaning the fire becomes larger and larger, spread faster and faster along the sample surface. We also look at the flame spread rate and flame length. You can see that on these plots, when we decrease the interbaffle distance, we see that both the flame spread rate and flame length first increase and then decrease. In other words, the fire characteristics has a non-monotonic dependency on the confinement level. And then on the lower right plot, you can see that at the same confinement level, the reflective baffle gives us longest flame, followed by black baffle, and then transparent baffle. Okay, in order to explain the the observed phenomena. We conducted numerical simulations. Here you can see some comparison between the experiment and numerical results. On the left top corner, we can see our real flame and simulated flame next to each other at different time instances. And on the right panel, you can see flame position, flame length versus time in experiments and in numerical simulations. So our model really nicely captured what we observed in the experiment. The benefit of having a numerical model is that after model is validated, the model provides detailed flow profiles. These flow profiles might be hard or even sometimes impossible to measure in the experiment. So with these detailed flow profiles, we were able to conclude that the effect of confinement on the flame, there are many different effects. Some strengthen the convection process, some weakens the convection. Because of these competing effects, we observed the non-monotonic dependency of the flame characteristics on the solid burning process. Okay. All right, so here's a quick summary of our project. For our ISS campaign, we total have 18 test days. 6 astronauts help us to conduct total 63 burning tests, and these tests give us more than 100 data points, and the comprehensive data set we built not only benefit our own project, but also can be used for future model and theory development. Here, I would like to really thank NSF and CASES for this unique opportunity, and I would also like to thank everyone who helped on our ISS experiments. This includes NASA JRC, Marshall Space Flight Center, Zing Technology, MSG team, and Johnson Space Center. And of course, we thank every crew member who helped us, and I appreciate all the support I got from the students at Case Western Reserve University. Thank you.
So thank you for those presentations. I'm just continually amazed by the breadth of research that's being done on the ISS and the potential impact to life on Earth here. So let's dive into the questions. Again, if you'd like to ask a question, you can submit them through the app. So this question is for all of you or any of you to answer. What lesson would you suggest if you were advising a first-time researcher on the ISS? Whoever would like to start.
Takes longer than you think it's going to.
I guess I would say the way you imagine it inside your hood or wherever you conduct your experiments is not how you would actually do it in microgravity. So there's this adaptation process, and I think working with an experienced implementation partner will help you translate or adapt your bench research into microgravity research.
Any other suggestions?
I would say probably you need to be flexible because there will be a lot of limitation or unexpected difficulty you may encounter, and then that not happening in your lab. In your lab, you can just fix it or whatever, right? So I think you need to have contingency plan or backup plan. Be flexible.
So with any of these challenges, or as we heard earlier, these are learning opportunities, have you been able to apply these to your work back on Earth, perhaps in other research lines?
Well, one of the lessons I learned in our work was that, essentially it was one big experiment, but it was really one experiment, and we spent maybe several years preparing for that one experiment, and I would probably say I spent a lot of time on that one experiment, and so I think that it was well spent because we had a very successful launch. And I think one of the things that I take back, to my other projects is really about the importance of being meticulous and being detail-oriented, and even the little things that we would not have normally thought about, like trying to simulate it using a rocker, for example, to simulate vibrational motion. We wouldn't have thought about doing initially, and that was actually a recommendation from our implementation partner. But I think the detail, the meticulous nature that we went about planning for this one experiment, like all or nothing, I think it helps us apply that kind of mindset to some of our other projects, even though it may not be all or none, but to improve our meticulous, approach to science.
Thank you. Anyone else have a?
Well, I think it really gives you an appreciation for the effect of gravity. So we've done a number of experiments based on what we were seeing up on the space station, and it really, I think it opened up a wide range of, a wider range than we may have considered in terms of the experiments that we're doing on the ground-based studies too. You take gravity out, and then you say, what happens when I put it back in?
True. All right, so a specific question for Dr. Chang. Your research found a set of 250 genes that are upregulated in space. Can you hypothesize why their production is increased in the microgravity environment?
Oh, thank you for that question. So it's more than 250 genes. Thousands of genes are upregulated. And I just highlight those 250 because those are functional genes that are important for hepatocyte function. I think one way to think about it, so the short answer is we don't completely know. And I think that's one of the exciting things about that tissue self-assembly and microgravity is an area of fundamental research where we can understand how tissues form, how cells interact with each other and form. But one aspect of it is that microgravity gives us a chance to take away the wrong signals, the confusing signals that we put on cells because we have no choice in the gravity environment. What do I mean by that? Well, hard plastic surface is actually a wrong signal, at least for a liver cell. For most cells in your body, they do not see a hard plastic surface. And we know from a lot of research, including some of the work from my lab, that cells sense stiffness and respond to that. And for the liver, it decreases their function. It's part of what we think makes the liver function goes down in fibrosis, which is the hardening of the liver matrix during that disease process. But another wrong signal is Matrigel. And the prevailing method of making organoids right now is Matrigel. And the Matrigel provides the wrong signal for liver organoids. How do we know that? Well, in the simulated microgravity vessels that we use, if we add back Matrigel to microgravity cultured organoids, the functions go down. And we think it has to do with, at least the gene signatures indicate, is a poorer differentiation. There's something in Matrigel that directs differentiation of these cells away from hepatocytes. And so microgravity is a way for us to actually take away signals that are wrong so we can add the signals that are correct.
Thank you. Moving to those great movies that we saw, Dr. First, what applications can you foresee that might harness the dynamic structures that you observed?
What form of applications could we see out of those dynamic structures, I think is a very intriguing question that we do not have quite yet. And one of the things is, why do we see those dynamics? So there's a whole area that's of interest in the physics community now from emergent dynamics and active matter. And it seems that we have perhaps another form of it. So I'm really kind of curious about that. And where that field is going, perhaps we can contribute to it. But the technological applications of these particular fluids, other than the nanostructure that I was talking about, the fact that you form microstructure gives you properties like a liquid to solid transition and that sort of thing. So you can use these materials for actuators and pressure sensors and all sorts of other applications too. So it's a wide range. Maybe I'm too far out in space sometimes thinking about this.
Literally. All right, thank you. So a question for all of you. What ISS payload constraints are most limiting for your fields of research?
Well, maybe, and you said most limiting, or?
Yeah.
I think one of the considerations with biological research has to do with the temperature stability of the different biological elements, from using the growth medias and whether or not those growth medias can be stored in 4-degree temperatures versus -20 versus -80, as well as the drugs that we're giving, right? These different small molecules, what kind of temperature requirements they have. And those ended up being constraints for us, because for the crew to be able to change the media, there's a process of needing to thaw out these samples and prepare these media for media exchange. And in the cases where the drugs need to be already in those medias, we had to determine whether or not these drugs would be stable if we just kept it at 4 degrees. So in other words, we can pre-mix the drugs, into the medias at 4 degrees, and whether or not those drugs could be stable at 4 degrees, for up to a couple of weeks without losing significant bioactivity. And for almost every drug, we had to sort of determine that empirically to see, how long of storage at 4 degrees were those drugs still functional. And I anticipate that if there were, technologies where these drugs could be stored at -80 or at a temperature that could still be equally convenient for the crew to be able to then deliver to our bioreactors, that would be a major benefit for future drug screening studies. But as is right now, some of the considerations are, can we adapt our potential drugs of interest to a 4-degree environment that are preloaded in reservoirs so that they could be just directly delivered, to our cells?
Go ahead, Dr.
The biggest limitation that we experienced was really the bandwidth of data coming down, which is a tremendous improvement from when we were doing experiments back in 2008 with Peggy Whitson. We have a lot more data bandwidth that we were able to monitor the experiments, but the real data, because our experiments, real time, if we could get that data right away, we would love it. We had to downmask the SSDs in order to really have the data that we could analyze. So that's an interesting, I think, limitation. And then it's crew time. They're busy, a lot busier than maybe a decade ago.
Yeah. So for fire, of course, the limitation will be fire safety concerns. So I have a lot of wild, idea, but say, for example, I want to burn battery, but it'll be very, very dangerous, very toxic. So as you can imagine, I'm pretty sure, if we really, really, really want to do that, we can construct a safe platform. But I can imagine it will be, a lot of people will be very nervous and propose to do that. Another thing is we really would love to see, how fire behave in different conditions, like including high oxygen environments, because we know future space exploration atmosphere will be enhanced oxygen 34% or something. But I think, from what I know, I think probably if I propose that, cases will not be comfortable. Let me do that. So I would say for fire experiment, the limitation will be safety concern, which is understandable, which is very understandable. Yeah.
I would add on that for biological experiments, a major limitation is the inability to bring back cells or tissues alive. And I think that because for a lot of advanced analytics, lab-based on Earth are still the better place to perform them. And the analytics on space station, although they're improving, are still limited. And also, overall for the field, the concept of biomanufacturing for the benefit of Earth really relies on us being able to bring back these tissues alive and functional.
So a big thank you to our amazing panel for sharing their extraordinary work and all that you're doing. It's been a pleasure to be on stage with you. This will conclude our session, but let's give our speakers another hand. Thank you. And so I'm going to toss this back to our MCs.
Oops, we got me on? All right. Awesome. Well, thank you so much. That was a very deep dive into the science right there. So thank you so much. And it's great to go out there and see and hear some of the people that I've had the chance to talk with in the past. So that, I guess, wraps up day number 1 for ISSRDC. Is your mind blown?
My mind is blown, and my eyes are tired. But I'm going to tell you this, it was a fantastic day. So some reflections I guess, for now? What did we learn about the things we did?
What did I learn about things? I learned first and foremost that there's a lot of people in here that are a heck of a lot smarter than I am, but I would also say too that I thought it was really interesting, especially when you were talking about, what it was like to try to do R&D not only in your lab setting, but trying to configure that into a space-based environment, and to have to work alongside various partners that are able to kind of take your science and make it something that is habitable on the space station. It really shows to me the amount of collaboration and the coordination that really goes into every single experiment, and to me, that to me is really exciting when you look at the notion of partnership and how it is that we all work in unison to be able to achieve a mission.
Yes, I agree with that. And just some of the other things that I learned is just getting inspired by Chell and Jessica this morning during the downlink, and just learning about some of the things that they've done in space and all the things that they've learned themselves, being a medical doctor, being a geologist, right? And then learning about what's going on in terms of space policy and figuring out what our leaders in the ISS program are concerned about, and some of the things that we're working toward in the future. So lots and lots of insights shared today, lots of things to think about as we move on to day 2.
Well, and what I think is also really fascinating about how we finished up here is we finished up almost exactly right in time for us to go straight into the reception. Strategery, so well done. So with that, though, I do want to acknowledge the fact that, again, at 6 o'clock, we will be having another networking reception for us all to come together and congregate and celebrate us being here in Washington, D.C. And then you will also have the opportunity to equally meet with some of our sponsors once again. So we do want to thank Boeing, the Conference Platinum sponsor, Orbital Reef, our gold sponsor, Northrop Grumman, Leidos, KBR, Airbus, Redwire, our silver sponsors, and of course, all of our marketplace sponsors. So with that, we'll call it a day, and we will see you bright and early tomorrow morning.
Awesome. Thank you so much.